Whew, that's a big slide. So let's look at that. So when we look at the eyeball's coverings, so the outside, we go from the outside layer inward and slice them and peel them off. We have 3 layers. We have an outside one, and they call that the fibrous tunic. Tunic means layer. And that is the sclera in the back. That's the whites of the eye. And mostly what that is collagen fibers that resist the stretch coming from the inside to give the eyeball a firm, not appearance, but a firmness to it that it can push a little from the inside and it doesn't just keeps expanding. So collagen bundles, collagen fibers are really strong when you want to stretch them out. And so the eyeball, therefore, is really solid to stretch. And the outside layer of anything is usually, fibrous and protective and anchors the organ into places. Of course, in the eyeball, that's not the case because we've got to move that thing around. So the sclera is not going to anchor it. And then in the front, when we get forward, we get the cornea in the front. And that's obviously not white. That's right here because that's the place that the light comes in. So that's got to be clear. But it's also very tough and very strong, and particularly very regenerative. That's also a very cool thing about it. It heals really fast. So that's the fibrous tunic. Then when we get below that, we get to the red one. That's the vascular tunic, and that means there's a lot of vascularity there. So there's a lot of blood supply. In the back, it forms what's called a choroid here. And the choroid is, besides the blood stuff, is a pigmented area that limits its light reflection inside the eyeball. So you visualize, light comes in here and light goes all over the place. And if it bounces off all the walls, it gets all scattered inside the whole eyeball. And we don't know what we're looking at. And so a lot of it's kind of like a movie theater. Only the screen is light and the rest is darkened, so there's not all this reflection. We can't really see what's going on in the screen. It's kind of like that. And so the pigmented area of the choroid is doing that inside the eyeball. It harbors blood vessels. We talked about that supply the eyeball. And in the front, what's really cool in the front, it has this ciliary body or muscle. And that controls the thickness of the lens. And the thickness of the lens is important because if we see something nearsighted, from close, the lens needs to be fairly round. We'll talk about why that is in a minute. And when something comes from far, the lens needs to be flat. And the way we control that size of the lens is that we have these muscles here. These muscles right here. That's the choroid. That's the ciliary muscle, the ciliary body, right in this area, right here. And suspensory ligaments, these little things, these little cords are attached to that muscle and they go into the lens. And it helps the lens accommodate when we do that. So when the muscle contracts, it's shortened and it pulls on that lens and it makes the lens flatter. When the muscle is relaxed, it lets those ligaments go and it makes the lens rounder. So that helps for accommodation. And then another part of the vascular tunic is the iris. And the iris is made of 2 muscles. The iris helps us create the size of the pupil. So we have one muscle, when the pupil is small, in pupillary constriction, we have the sphincter muscle, the sphincter pupillary muscle, contract. That's a round thing around here. And on the other hand, when we want to make the big pupil, and by the way, that's a sympathetic reaction, then we have dilator muscles, dilator pupillary muscles, that are radial, they are out here, and they pull that iris close, and therefore the pupil gets big. And that controls the size of the pupil, regulating the amount of light that enters the eyeball. Whew, almost. We got one more tunic to go, and that's the internal tunic. And that's the retina. That's where we actually see. The retina in the back has photoreceptors in it. So that's the yellow stuff. So back here, we got photoreceptors. And photoreceptors are here to make electrical impulses out of light. And so when the light comes in, it hits the photoreceptor, imprints itself on that area like a screen. And when the light hits, those cells fire off and send a nerve impulse to the brain. And voila, we can see. So the back is photoreceptors. We also have pigments in there. So we have a lot of pigmented areas also within the retina to make sure the light doesn't get scattered around. It only goes to the place where we want to transduce it. And then we can send a nerve impulse to the brain. Got it? I know, that's a lot of stuff, huh? But we are not quite done yet. In the inside, the eyeball has two chambers, because what we could do now, we slice it and we'll look at the inside. The back of the lens chamber is one of them. The front of the lens chamber is the other one of them. We have 2/3 is about in the back, and that's filled with sort of a glassy, gelatinous, thin, watery stuff. It's mostly water, but it does have collagen and also hyaluronic acid in it. That thing is just made and it stays there. The vitreous humor, they call that. And that thing helps put pressure from the inside out to the eyeball and stabilize the eyeball, that we don't just squeeze it like it's a flat ball. You know, don't let the air out. Don't let the vitreous humor out. Then in the anterior chamber, in the front, lies the front of the iris, front of the lens, lies the aqueous humor. And aqueous refers to watery. So that's more of a watery substance. And that provides nutrients to the lens, needs a lot of nutrients. There is muscle function going on that needs nutrients. The cornea needs to be repaired. That all needs nutrients. So there is a lot of stuff that needs ATP. And when we need ATP, energy, we need nutrients. And so the aqueous humor gets secreted by the ciliary epithelium, they call that, right around this area here, the ciliary body. And then it moves around and it actually exits through what's called the canal of Schlemm. And so then we have a continuous flow, so we can continuously bring nutrients in. And we also do maintain the internal ocular pressure with this flow, so we can decide how much fluid goes in and how much goes out. And if we have an intraocular pressure rising, because maybe these channels are blocked or something like that, then we can get damage to the retina in the back because it's too much pressure for all these sensitive cells back there to work, in the back of the wall of the eye, back here. And we call that glaucoma. And so we got to do something about that, because we don't want that cause us blindness. We don't want blind. That's what we don't want. This brings me to the eyeball movement. Oh, look at that. I cut it off. Oh, well. The eyeball movement is very delicate. You got to move that sucker around a lot to read, to do everything. You got to move it fast. So actually, from a size strength perspective, these muscles are extremely strong. And we have 6 of them. We have 4 that go in on the side. Here, they go to the side, they go straight back. Oh, and they actually, what they do is they anchor in a ring back here. Like it's called the Tenderness Ring. And that attaches into the bone then. So the muscle goes from the eyeball, the sclera, to the ring, and then the ring's attached to the bone. And so we got 4 on each side, just going straight back. And those, you know, when you look at what they do, is they pull the eyeball, the medial one, or the lateral one here, the outside one pulls the eyeball to the outside. The one on the inside pulls the eyeball towards the nose, to the inside. The one on top brings it up. On the bottom, brings it down. And then, though, then we got 2 triggers. Oh, see, here's the motion. I like this picture. I put the motion on. So you can see the arrow here. The motion goes up, motion goes down, internal and external. But then we got these 2 suckers. So we got one of these muscles, like the superior oblique here. Look at that. That attaches right here. I know I should take a different color pen, huh? Attaches right here and goes back. And then on the inside there, by the nose, there is a little loop. It's called a pulley. And the muscle goes through that pulley and then goes to the back and anchors in this back ring. So that's kind of goofy. So that's called the superior oblique. And I know you would think that thing wears out, but it actually doesn't. And then we got an inferior oblique, and the inferior oblique muscle is attached on the lower orbital rim. That's right in here, and then it goes to the, it goes from the outside. It goes from the inside of the sclera, and then it attaches into the inside of the orbital ring. Arises from the lower orbital rim and attaches into the outer side of the sclera. No, I gotta review that. I will call that wrong here. Inner side of the sclera. But anyway, let's talk about the motions that they do, because that's the goofy part. When you look at this motion here, the eyeball actually is pulled down. No. Down and out. Sorry about that. I know, I always get it wrong too. So when you look at that, the eyeball actually goes down and out when that one is pulled. So this here, I like the way that this is describing it, because it sort of shows how it rotates it this way. When we do the inferior oblique and we work that one and we we contract that one, it pulls the eyeball up and out. So it pulls it sort of this way, basically, and we gotta stare up that way. I know, we're gonna have to do that in class with the eyes present, because that's kind of a difficult situation right there. But let's keep going with further in and move away from the muscles. Let's go to the retina and let's see how we actually gonna hear. So the retina is the light sensitive area, a photoreceptor layer, and it contains 3 levels of neurons. The bottom one, so here, back here, back here, that's the retina. That's the back of the eyeball. That's where the retina is, right here. The first layer of cells are right here. There are 2 types. They're rods and they're cones. And they're embedded in the back of the wall. And then they attach to the next level cells, and those are called the bipolar cells. See here, it says bipolar cells. So these orangey looking ones are the bipolar cells. And then from there, we got a few more cells. We got actually much more than that, but we got some horizontal cells here. They're in between that layer. And then we got some other ones here. They call them amacrine cells. They're right here. They're also in between that layer. But then from there, we get to the third layer. The third layer are ganglion cells. And then they collectively get together and exit as the optic nerve, and become the optic nerve and exit the eyeball. Now, what's interesting about the rods and the cones in here, when we look at when light hits it, inside of it, we have these disc things. See these here are the discs, and they're stacked on top of one another. And inside the membranes of those discs, we got this rhodopsin molecule. And the retinal part of that molecule, right here, is shaped with a kink. And when light hits it, the kink straightens out. I know. Don't get depressed. But that's how it works. The vision basically is a molecule straightening out when light hits it. And we just have a gazillion and gazillion of these receptors in there.